Hey everybody, this is Miss Aninga coming to you to help you out with your importance of shapes and size of cells lab for biology. So today what I'm going to do is I'm going to set up the first part of the lab, which is looking at the importance of size to your cells. So right now what I'm going to do is I'm going to be taking two different discs here, two different sizes, and I'm going to be soaking them in vinegar, which I have here. Now that vinegar is going to cause this to change color because there's a base in here and the vinegar is obviously acid, and there's also a pH indicator in there, so it's going to change color as it sits in the vinegar. Okay, but before I start that, I want to take measurements of my discs. So I'm going to take my big one right here, take my ruler, and we can see that the diameter of this thing is just about 4 centimeters. So our large disc. Oh, pencil, why? It's okay, I got an extra. Our large circle, or our large disc, has a diameter of 4 centimeters. And because it's a cylinder, it's not all the way rounded. We're going to look at how deep it is, and it's about half a centimeter thick or high. So 0.5 centimeters. So since I cut these two discs from the same, larger disc, our smaller disc is going to have the same height, but it's going to have a different diameter, as you can see, because it's smaller. So we're going to take our small one, and I'm going to switch places with the big one so that I can have a flat surface to go on and measure, and we can see that the diameter of this one is about 2 centimeters. So it may not seem apparent right now why that is important, but I promise I'll do the math in a separate section here with you in a little bit. So this stuff is important to remember for now. I'm going to move us over to the viewing area, which is over here. Alright, so as you can see, I switched this around because it was a little bit too bright. So got my discs here, and I got my timer, so it shouldn't take very long. So my stopwatch here on my phone should do just fine. And what we're going to do is I'm going to put that vinegar in and see how long that's going to take for these to change color. What you should do on your sheets or on your assignment, on your lab assignment, is to predict which one is going to, completely change first all the way. Okay? So I'm gonna add my vinegar until they're both covered there and start. This might take a bit, so if you end up, just skimming through, that's fine. I'll probably end up fast pacing this so that you can see it better, and then I'll probably go over the calculations for why this is important in another part or another video. So, alright, see you then. Alright, I'm gonna stop there, and you'll notice there's still some blue in this little one here, but it looks like that just means there was an inconsistency in here. The moment that it was like completely like, once the color change reached the middle, that's where we should have considered this one done. So just keep that in mind. But yeah, that was a quick like way to see how they change and which one changed faster. So there was my time again. Okay. So, I'll be taking a look at which ones, why that's important here, in the next section here by looking at the math. All right.